I'm Tom Zhang, and this is our team. I will let everybody introduce themselves, but I just want to say what a great opportunity for us to get together. It's been so many years that we worked on this, but we don't get together very often, right? We just talk to each other all the time, but we actually don't get together that often. And one of the reasons is because I'm in China, 90% of the time. But this is a great opportunity. So let's see what we can do. I just want to give probably the latest update. The latest in China, we are getting some attention. When I get back, maybe I'll get on the China Central Television to be interviewed by one of their top investigative reporters about the project. So it's getting interest from higher up people in Chinese government, because one thing we know is that the Communist Party of China likes to take things from the rich and give it to the poor, right? So this is a good project where people who are poor can get something. And so the paper we finally got published with lots of help from our Chinese friends. It says that the treatment, Warburg treatment, is 100% safe. So we get a chance to implement it everywhere. So we're hoping to do it here. So maybe Mike can start talking a little bit too. You don't get the chance to talk too much.
No.
Yeah.
Sure. I'm Michael Badowski. I got my degree in microbiology and immunology with Tom. He was my unofficial mentor, and he taught me most of my great lessons that I learned about science and about laboratory life. And I feel honored to have learned a lot from him, from you. And I've said this to anybody else that ever listened to me, I don't think I've ever met anybody that had more vision than you. And that's what really drives this project. So if it's not been said, clearly, we all appreciate the drive and the vision that you bring to solving this problem.
Well, thank you. And, I want to have this on record that Michael Badowski is the only person I would openly admit that is more creative than I am. so I would admit that every single time. And Michael Pennington here is someone I've said, 100 times, knows more about cancer than I do. And that still is true, even though I keep learning. And Linda has been not only a teacher, mentor, a friend, but an inspiration since early days in my career. When I was a student, I was pretty lost in what do you do with this degree? What do you do with this knowledge, right? But Linda was the constant. Just do a good job, do good science, and then do the best you can. And what we're doing is just science. Here's something we made observation of, and we want to share it with the whole world. And scientists are supposed to publish, right? You're supposed to communicate, right? But from time to time, you have something that the world is not quite ready for, but it needs to be out there. And so when Mike hears, credit me with vision, I'm really just doing what every scientist always do, which is to just tell people about what they have learned and share with everybody about with the knowledge.
Well, I think one thing that people, sometimes we say are not ready, the world's not ready to hear, is sometimes I think the world is not ready to embrace an old idea. If it's not new, if it's not the sexiest thing, if it's not the most current thing, and what Otto Warburg accomplished decades and decades ago was to lay some groundwork for something that had a really good scientific basis. For you to bring us in and recognize that this is something that needs more attention, I think is really interesting, really important, and like you say, it's just good science. But that's part of the difficulty is it doesn't have the cachet of the newest thing, but it does have, the more we learn about it, the more we learn that it does make sense. It does pass the scientific smell test. It does, it vibes with everything we know about cell biology. It vibes with everything we know about cancer biology, about immunology. And he might not have known that decades and decades ago, but now we know a lot more, and it still makes sense. And that's what's exciting about it.
Yeah.
Introduce. Let's Linda introduce herself.
Yes, Linda, take some time.
I'm Linda Mead Tolan, and I'm pleased to be here after originating in London, West Virginia, a town of 300 people. I am a biochemist who received my doctorate from the City University of New York. I was one of the first persons in the CUNY doctoral program in biochemistry. I ended up out here in Arizona, and I think that my background and my exposure to different things during my graduate career made me very receptive to innovative ideas. And I too consider Tom a visionary. I consider it a privilege to go on this journey with him. And I am pleased to see all the things that might be possible from this innovative idea that came from Otto Warburg.
Yeah, I think Linda is the first person that I actually discussed in depth about the science, and we talked about all the science, and you're the one who brought up this is glucose deprivation therapy. This is glucose deprivation therapy, and everything actually falls into place after that, because the world doesn't have a glucose deprivation therapy to treat cancer, and wow, it really needs one. And when we implement it, well, that's a good therapy. So
it's good to have a name that's accurate.
Yeah. Well, again, that's science. If you name it incorrectly, then the science kind of doesn't follow. And we've been calling it Warburg treatment because in 1927, Otto Warburg published an article, and he basically says use regular insulin to control the blood glucose, lower the blood glucose. You can treat cancer. You deprive cancer cells of glucose. This is glucose deprivation using Otto Warburg's idea.
And Otto Warburg was a very famous scientist who was nominated multiple times for the Nobel Prize before winning it.
Yeah, in my research, he was nominated 47 times. And the only reason he didn't win more than one is because he's a Jew in the middle of World War II, so in Germany. So that may make collecting the prize a lot harder. So.
Yeah, I'm Michael Pennington. I was at the Cancer Center for nearly 2 decades, and before joining Tom as his senior scientist in his pharma company, and I helped him do all the sorting out the science, optimizing the protocol, and figuring out exactly how we can make this a viable therapy.
Yeah. The specific again, scientific insight that Mike had on glucose deprivation will cause cancer cells to lose their ability to run multi-drug resistant protein pumps. That was something that Mike, just brilliant, brought up. This is a, great explanation why this therapy works. Is well, I'll let Mike do more explaining, but
Right.
this was something Mike did.
But why don't you have a conversation about how it started? How did you guys, who started this, how it, give a history lesson, however you want to do it.
Well, this is, I can't even find a starting point because we've known each other for so long. And it's more like this all started with discussion about scientific ideas but I want to get back to Michael Pennington is that, when he started thinking about what's the mechanism, why this works, why this should work, we were able to make it work better because of that. When you have a good hypothesis on why something works, you can tweak the protocol. You can make it work better, and that is so important because the protocol we use today is so reproducible. It's so easy to use, and the barrier to start is so low that we believe that this is how you make an impact in the world. Is you make the therapy available. You don't have to go to the big world-class cancer center to get it. You can come to a small hospital and get it, and people can learn it and do it 100% safe easily. And it's all because you understand the science behind that.
When we first got together in the laboratory, Tom and I, the chief scientific officer of the Cancer Center, a person who we respect a great deal and who we checked every step of our thoughts with, had him go over it, what we were doing, critique it, approve our experiments. So the chief scientific officer of the Cancer Center at that time was a renowned scientist named Dr. Tim Bowden. And we were all at the university, but I first met Tom when I joined Tim Bowden's lab as a postdoc, and Tom was a graduate student there. And it was really quite a training ground because we were exposed to Dr. Bowden's very objective, no nonsense. What experiment would you do if you figured that, if you give us a hypothesis? He was an excellent mentor and an excellent trainer, and he gave us very good guidance.
Yeah, that to me, he's the standard. And in the back of my mind, I'm always thinking, what would Tim think about this? You don't want to do anything that he's gonna say, well, this is not good. And one of the first things he's always going to say is, have you repeated this, right? Have you repeated this? And that's a pretty good start when you're talking about good science.
We'd be very anxious when we would get our data and take it to him to see whether or not he would, but he was always gentle, even when he critiqued it. He was always kind. He was also objective. He was always suggesting things that might make the experiments better. You need to do this control maybe, or something like that.
Yeah. And then Mike Pennington worked next door.
Yeah, actually, he was in our prostate program group for a while. Then when I was working at the Cancer Center, come to our lab meetings, and you become a much better scientist when he's in a room and you're giving a lab meeting.
Yes.
He's such a good guy, though. Yeah.
Yes.
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 Oh.
Why don't you talk through the process of how you started, what your original premises were, and how you moved along to refine whatever you did to get to this point?
What's your official story?
Well, it has to do with family, and
Have it self-contained. We started this process, so that's a full statement, not me being involved.
well, we started this process because in my case, my aunt, so my mother's younger sister, was diagnosed with breast cancer, and this is 2006. 2006, and we had to help her because she didn't want to get the standard treatments. She had seen her older sister die after standard treatment for breast cancer, and what it really did is that it forced me to start thinking outside the box, to say what can we do to help. And we came across this treatment called IPT, insulin potentiation therapy. Insulin potentiation therapy is part of the alternative treatment, and that's widely practiced and it's been around for a long time. But it's difficult to explain why it works, because if insulin is doing all of these things to cause the treatment to work better, then we have a brand new mechanism. And so what happened is, by letting my aunt doing IPT, I was able to learn about IPT. And it was, I think, the year after, Linda, after we have discussion about IPT, went to the IPT conference and found out what the IPT world is doing, and this IPT really became the predecessor of what we do right now, what we now call Warburg therapy, because in the beginning, again, Linda realized that this is glucose deprivation therapy that's being done. It's not so much insulin causing the treatments to work better, but rather the glucose deprivation that causes the treatment to work better. And ultimately, when I got to China, we had the opportunity to experiment on that. And we use insulin to deprive cancer patient of glucose. We lowered the blood glucose. And the more we lower the blood glucose, it seemed like the better the treatments are. Yeah, and so again, this is just bring the history back in, and eventually we realized that this is the correct name for this treatment is really Warburg treatment, because Warburg was the one who's been working on glucose deprivation, and insulin is just the tool, and because of that, we changed the protocol, we changed the method, and the method then is focused on glucose deprivation, and Mike is the one who correctly says, the lower you can get the glucose and the longer you can keep it in hypoglycemic state, the better the treatment is going to work.
Yeah, so IPT was an older protocol. It was based around some old hypotheses, that didn't stand up with scientific scrutiny over time. And the biggest issue is that they wanted to mix drugs with sugar, and they didn't quite understand the whole hypoglycemia is starving cancer. They didn't grasp that part. So their protocol was very inefficient, if not was wrong. But we were able to look at that and say, okay, but the insulin can do this. It can actually lower blood glucose. And if you just keep it low during the treatment of chemotherapy, you deprive cancer cells of its much needed glucose. that they use to survive cancer therapy, specifically the oxidative stress. These cancer drugs cause cancer cells to go in apoptosis, cause them to have drug resistance. So, yeah, it is a matter of how low can we go that's still safe, and how long can we go with hypoglycemia that is still safe, and how much drug we can give that is still safe. There's a lot of people out there that are terrified of low-dose chemo, and they should be. It is poison, it's high in side effects, and oftentimes it kills people faster than the disease itself. Now, there are a small percentage of people who respond very well to chemotherapy, but everyone else, no. All they do is get poisoned. So this method really, by lowering the blood glucose, actually makes the cells more receptive to cancer drugs. So you have a lower dose, less side effect, with not the same efficacy, more or less. You still need a full clinical trial, a non-inferior clinical trial with low-dose chemo that show this. But we actually do have very good responses from people that take it.
Yeah, and I also want to credit Mike Badowski on understanding the immuno aspect of this treatment, because one of the things he correctly, I believe, point out is that this is a great immunotherapy.
Right.
Yeah.
And that's really, the one step, and then the next step when we realize this is probably one of the immunological mechanisms that's taking place is if you can kill a small number of the cancer cells right at that point of therapy, it allows the immune system to recognize that there are cancer cells that can be fought. Those proteins are going to be exposed, the immune system is going to recognize it, and high-dose chemotherapy does unfortunately a great job of killing the immune system. And low-dose chemotherapy keeps the immune system intact to allow that self-immunization to happen, and then the immune system can go to work. A lot of people don't recognize that cancer is such a slow disease, by and large, months, years, and the body's just slowly losing the battle. And if we can make a little bit of a tweak while keeping the immune system intact, then a lot of good things can happen therapeutically.
Yeah, so this is actually observed in radiation therapy, where a tumor that's irradiated, a localized tumor, you actually can get an immune response to more widespread tumors that go on. And the reason for that is that the radiation's not systematic and doesn't destroy the immune system.
The standard chemotherapy works best on rapidly growing cells. Well, you've got rapidly growing cells all over your body. So the toxic effects of standard chemotherapy are inherent in the type of drugs that have been developed for so many years.
Yeah, I mean, again, I credit Mike Badowski for bringing in great immunology to this. So, and I also am going to freely admit that Mike knows more about immunology than I do. Yeah, and I think over time, because we developed this for late stage patients, and when you bring keeping the immune system healthy as part of your weapons against cancer cells, the quality of life is much better. Yeah, and it's scientifically much better science. And the way I explain it to patients and patients' family is that what we're really doing is that we're creating a tumor vaccine right inside the person's body.
Exactly.
And the reason standard chemotherapy you can't do that is, what Mike said, you use too much drug. If you can use small enough dose, then killing cancer cells, just like using radiation to kill cancer cells, actually causes the immune function to go and do what they're supposed to do, which is find more cancer cells to kill.
But when you use large doses, the immune cells themselves are rapidly proliferating cells.
Yes.
And so you're basically having toxic effects on the whole immune system that normally keeps the constant barrage of circulating tumor cells in check.
Yeah. And, we have these discussions about in the immunotreatment of cancer area, we'll call tumor microenvironment, where you have lots and lots of acid being produced by cancer cells, the fermenting glucose, the famous Warburg effect. And acid is not good for immune function. And when you kill off cancer cells that produce a lot of acid, you improve the tumor microenvironment. And everybody who's working in the field of immune therapy of cancer knows tumor microenvironment is a very big problem.
Yes.
But we believe this is a good solution. And Mike Pennington made a beautiful drawing, a diagram of that to show exactly how that might work.
Yeah, another thing is that the immune cells in such inflammation, they too need glucose. So you have these cancer cells and these immune cells. The cancer cells are out to compete the immune cells. For glucose, unless you go after those highly glycolic cancer cells, you can knock some of them down. The immune cells can get in there and have a little bit of their own sugar to fight the cancer.
Yeah. And, when we lower the blood glucose using insulin and keep it down for a while, it helps flushing out all the acid. Because you reduce the amount of acid, the circulation actually would help the microenvironment, which, is another enhancement of the immune function. Okay. So, Mike, maybe you can give a 2-minute summary of the treatment, since you're the kind of the mastermind behind the treatment details.
So what happens to initiating treatment? The initial phase is to give insulin. The idea with the insulin is that you're going to lower the blood glucose. And insulin, this is a very metabolic hormone in the body. The pancreas makes it naturally, and it causes uptake of glucose, primarily by liver, adipose, and muscle cells, an induction of insulin. So your whole body is going to suck up all of the glucose when you're given insulin, or your pancreas produces insulin. And it's usually in response to eating a meal per se. But in this case, we're giving exogenous insulin through intravenous, through an IV.
IV.
Yeah, intravenously. And you don't give a whole lot of it, depending on the person. Some people need more than others, but you give usually around 10 to 20 units for a normal person, and their blood glucose from there was dropped out. That creates a hypoglycemic treatment window where you're able to treat cancer cells with doing chemotherapy drugs. Now, we typically use cancer therapy drugs, although we use many others depending on cancer and whatnot. But the first one you can use is cyclophosphamide. This is an old alkylating cancer drug, an alkylating agent, so it causes oxidative stress within the cell. And this is a prodrug that gets converted in the liver. It's a little more long-acting, so we give that at the beginning of the therapy because it's about a 20-minute lag for it to really ramp up and start affecting cancer cells. And then we also give 5-fluorouracil. This is a folate inhibitor, and it interferes with more of the metabolism in the cells and DNA replication. And these three drugs, pharmacology, get the cells harnessed when the blood is up. Blood glucose is at its lowest.
All right. That's a great summary. And the fact is, we tried different drugs, and these two seem to work extremely well together and have very low side effects. And maybe Mike can say, what does it feel like to have the blood glucose really lowered? I mean, what it was.
Right. So I have my own cancer issues. I have a immunoblastoma. It's a cancer of the tooth, actually. It's a immunoblastoma uses tooth anatomy, and they're really from a tooth anatomy from their toothpaste commercial. And it's a very locally aggressive malignant cancer that grows up from where the tooth is and all over. It's been a problem for me since 2011. I've been on Warburg therapy on and off the treatment since 2019, so 6 years now.
Yeah.
And 6 years ago, it was declared terminal with a very poor prognosis, so I'm still here.
Yeah.
And I'm using a combination with a targeted kinase therapy. So what happens is, you get the IV put in, you have the insulin inserted through the IV, so you get insulin. And it really takes a good 20 minutes or so, at least, for the blood glucose to go down. And you get a little sleepy. Some people get a little sweaty. But it's actually rather mild. It's not a big deal. It's incredibly rare that anyone has an issue with it, right? I mean, where they need, panning on the face with sweat or something and stuff. It's actually quite mild. You never go so low that it causes anyone any real issues. I mean, you never go long enough that causes anybody real issues. So it's actually very safe. And we have a safety paper now with cancer medicine that attests to that. And then the cancer drugs get put in, and you just kind of sit there in the chair when the cancer drugs do their thing while you're in hypoglycemia.
Yeah, what's really nice about these two drugs is that their half-life are short, especially
Right.
5-fluorouracil,
Right.
16 minutes.
Yeah, so we went through there and we figured out the exact timing of these drugs, half-life, pharmaceutical half-life. These are very well documented. Their own drugs are very well documented. So you sit there and figure out the precise timing of when to give the drugs so that they have their peak efficiency during the lowest point of hypoglycemia.
Yeah, and so at that 20-minute, during that 20-minute, when the blood glucose is very low, there's actually quite a bit of drug there. And but with the short half-life means an hour and 20 minutes later, the 5-FU is gone.
Right. And the same thing with the insulin. Insulin is the natural human homolog insulin. This is the long-acting pegylated version. That's what we're using. This plain old insulin. That's a half-life around 4 minutes or so. It's very quick acting. It doesn't stay in the blood too long. So if we ever had an issue, we could just let IV glucose.
Yeah, and we do
That
that.
completely raises blood glucose right back up. And any ill effects of hypoglycemia go away.
Yeah, we do
So
that.
this is actually,
Yeah.
the hypoglycemia part is actually very safe. The doses we're using with the chemotherapy drugs, they really don't show up on blood work. Now, we've had patients in the past that had good dose chemo, and their blood work, they come in, their blood work doesn't look good. It has a hard time coming back because their bone marrow has been so damaged by the high dose chemo that they really aren't going to have a good blood test.
Yeah, these two drugs have.
And the dose we give them, that does not happen.
Yeah.
Your blood levels are always fine. The CBCs always come back perfect for me.
Yeah,
It's never an issue.
these two drugs have both been in clinical use for about 60 years. So every doctor knows how to use it. And that makes teaching it much easier because, the new cancer drugs were pretty complicated, what to use. And these are very simple to use drugs.
Yeah. And that speaks to the safety of the whole process.
Yeah.
Insulin is well known, drugs are well known,
Already
well
approved.
documented,
Yeah.
and generations of scientists and physicians have already been using them. That
Yeah.
makes everything that much more, I guess, stable, right? For everything we know about the therapy.
And traditionally, in analyzing the effects of different drugs, the maximum tolerable dose has been determined, and the idea may be that more is better, and more will really knock the cancer out faster. But more also knocks out all of the rapidly proliferating cells better as well.
Yeah, this is the exact opposite, whereas what is the minimum effective dose? Right? We're not trying to kill the cancer before we kill the patient. Can we just alert the immune system that there's cancer?
Yeah.
And
What's the minimum it takes to alert the immune system?
What's the minimum?
Yeah.
Yeah, it's the minimum it takes. And this therapy seems to hit all those marks. Huh?
Yeah. The nice thing about this therapy, because it has these little side effects, that you can actually put other therapies on top of it. So you can take a immunotherapy, such as a antibody checkpoint inhibitor, or you can have some other targeted antibody or immunotherapy or even a kinase inhibitor therapy.
Yeah.
In addition to this chemotherapy, without ending up in the hospital.
Yeah, in China, hospitals, the patients routinely get Chinese traditional medicine, herbal medicine, and there's no problem with that at all, to combine them.
Well, and can be repeated more often.
Correct.
So since there's not huge damage to the patient and to their immune system, a person can easily handle multiple therapies at a schedule that traditional high-dose therapy can not even dream about.
Right. So term that is a fractionated dose therapy, where you're receiving a fractionated dose on day 1 and day 3 and day 5, and repeat that for 3 weeks. And in that 3-week period, you can actually receive the same amount of drug as your traditional chemotherapy regimen gives it all in 1 day.
Yeah.
And you still have that low side effect profile.
Yeah, you have low side effect without using truly low dose. You just you can have the same amount of dose, but not as much side effects because of that. And what about the cost? The cost is really what got this treatment going in China, because China has this poverty alleviation program that during the last 40 years raised 800 million people out of poverty. 800 million people, and it's a very powerful program. And this got the support of the poverty alleviation program, because many people, if they go through standard cancer treatments, they're going to be bankrupt. Yeah, or and was this because the cost is so much lower? It's very popular. People can choose it.
And that has been so, the capacity for people on chemotherapy to go bankrupt has been documented in a paper co-authored by several members of the Cancer Center and their colleagues, including the person who was head of the Cancer Center at one time, that 47.2 or 0.4% of people who undergo traditional therapy are bankrupt in 2 or 2.5 years.
Yeah, Dr. Alberts.
Dr. David Alberts.
Yeah, it used to be 25 years ago, 30 years ago, the annual cost for cancer therapy is in the $20,000 range. That doesn't even cover 1 month of therapy today. So yeah, the costs have skyrocketed immensely.
And that's because it costs billions of dollars to run it through the system as it has been set up. When we went to our colleagues to ask them how they felt and critique our science, because as scientists, we wanted to make sure that what we were doing was scientifically correct. They said, "Oh, that's fine, but you need to do clinical trials." "Oh, that's fine, but you have to go through this particular clearance or that particular clearance." And that's fine, except we didn't have any money.
Yeah. Well, actually, we don't need to go through clinical trials because we're using already approved drugs at already approved dosages. And it's up to the doctors and then the patients to agree on that. And especially since we do it for palliative care, this is not correct that this needs clinical trials. And of course, we'll welcome anyone who will support a clinical trial. We'll be very happy to do that. And I'd love to see a side-by-side comparison of the Warburg treatment with standard chemotherapy. I'd love to see that.
Traditionally, scientists develop their results by looking at a system that's gone wrong and figuring out how to make it go back to normal again. And in this case, we are trying to keep the system from going wrong. This technique has the possibility of opening up a lot of areas of cell biology and posing a lot of questions. One of the big things now in cancer research is the role of the tumor microenvironment and what contributes to dormancy in tumor cells and what makes them come out of dormancy, because it's been found that tumor cells can lay dormant for long periods of time for unknown reasons and then come out of dormancy. So by using this technique, I look forward someday to being able to do research to use this technique as a tool for understanding more about cell biology.
Yeah. And, I just want to address the path we have taken. This really got traction because we started to treat poor patients. And poor patients who have no money and no choices, they love this. And but in terms of science, I think a great combination therapy going forward is actually with the immune therapies of cancer. I just think that this will make the immune therapies work so much better.
And how could it not? I mean, there's been so many immunotherapies that have been developed over the last 20, 25 years. And some of them work well and some of them less well. But a lot of the heavy-duty immune therapies, we're talking, CAR T therapies that cost $600,000 for a treatment, $800,000. We know there's no realistic economic usable future in that. The science is solid, but that's not feasible.
Well,
What ways can other immunotherapies that have medium efficacy now become far more effective in combination with Warburg therapy?
Yeah.
And that's really part of the question. It's funny, you're thinking, it's like, what are the questions we can answer in cell biology? And of course I'm thinking, what are the questions we can answer in immunology?
Yeah.
Because that's
It's a tool. It's really good hammer.
Right. My hammer is immunology, your hammer is cell biology, Mike's hammer is cancer biology, and we all have our areas of interest. And we're trying to figure out what's going to work.
I also think we should talk about PET CT scans. I think over the last 20 years, the gradual increase in the usage of PET scans changed how people feel about glucose and cancer, right? So, PET scans really start going, I think, early 2000s. Then, you start seeing it. And in China, it's still relatively rare, but everybody knows about it. And here, I think, it's standard for detection right now. But the thing I talk to people about is that the Warburg effect, we call ourselves the Warburg treatment. The Warburg effect is used during detection of cancer.
It's this hallmark of detection.
Yeah.
It's the gold standard.
Yeah, it's a gold standard. It's the best there is to find cancers. But during treatment, the standard protocols right now pretty much ignore the Warburg effect in treatment.
Yeah, this is a gold standard, has the highest specificity and sensitivity for finding metastatic disease. When you're talking about FAT, deoxyglucose PET scans, that's a specific tracer molecule. This is a derivative of glucose that's used in these scans. And what happens is the cancer cells absorb this, and it's not able to metabolize it. It needs a slightly off-kilter sugar molecule, abbreviation of glucose, and it accumulates within cancer cells. So it can show up on these PET scans very efficiently. So when a lot of times, as you hear in the States, when cancer patients come into cancer, one of the things they do is they go get a PET scan just to stage them to figure out how widespread is the cancer.
Yeah.
So for diagnostic purposes, this is standard practice. It's not standard practice for treatment because we have a hard time pharmacology treating glucose in cancer cells.
Well, people are looking for something, but
Right.
you know,
It's a gold standard for location, but it's also a gold standard for the degree of malignancy of the
right?
tumor in that area.
Yeah.
And so it serves a dual function, not only where it is, but how bad it is too.
And we see that's associated with pain. When you have someone who has a PET scan and shows a spot that is taking in massive amount of glucose, almost inevitably pain is involved. And
The interesting thing about the PET scan, there's three other things that really light up. One is the bladder where it gets clear, and that's not relevant. But it's also the brain and the heart that really need glucose as well. So it's very hard to interfere metabolically with like we say, a small molecular inhibitor of glucose without affecting the heart and brain. And that's actually not what we're doing. All we're doing is lowering the blood glucose. So there's still enough blood glucose there for heart and brain to function perfectly fine and safe. But these cancer cells really do light up. Linda's book, they really light up. The more aggressive they are, the more they light up because the more sugar they use. They really need the sugar. And if you take that away from them, then you create the vulnerability.
yeah. So when I explain to patients and families, I talk about PET scans. This is why this works, and the principle. I think everybody now knows that glucose and cancer is closely associated. And what's interesting, I mean, if you go back to the 2001 article, the original Cell Super article on hallmarks of cancer, this wasn't discussed.
Mm-hmm.
This was not one of the original hallmarks.
That's right.
That's in hindsight, it's shocking that this is how we find cancer now. But yet in 2001,
the PET scans.
Yeah,
It's very important.
And to use only dietary methods to decrease the glucose or carbohydrate is not going to be as effective as using a short-term temporary method like this because of that brain-heart connection. You can't go on a diet and be on a diet for a long time with glucose so needed by the brain and heart.
and it's also hard to do in scale. I mean, how do you help lots of people? Yeah, patients.
Patient compliance. Yeah, you're not doing that for most people.
Yeah.
There are very few people out there who have the will or desire to not have any carbs in their diet whatsoever.
Yeah, and they don't know how to do it.
Gluconeogenesis, the liver that can produce its own sugar.
Yeah, but people wouldn't know what to do.
I say that because so many times we've been asked, well, if I just cut out sugar, will that help be effective in preventing cancer? And you can't cut out all sugar because you need it for your brain and your heart.
Yeah, and it's very difficult to do.
Yeah.
Well, it's really impossible. The homeostasis will only get you so far.
Yeah.
And so, the insulin, we need to push that down just enough then to, like you said, to kick that vulnerability into place. But what's also interesting, go back a second, we talked about, pain in the patient is really an important issue. I mean, this is designed to be palliative care.
Right.
And it works in that fashion because we know that advanced cancers, a lot of pain with it, and it's something that has certainly been observed is that pain decreases in these patients.
Yes, it has.
And you've seen more of that in these patients than anyone.
It's reproducible.
Yeah.
And it's highly consistent.
Hundreds and hundreds of times.
I've seen it by far the majority of the times that we tried it.
Yeah.
And you don't have to have 54 treatments to see that effect. You can see that effect within the first 4 treatments.
Most of the time.
Often.
Yes, most of the time. That's actually the part that's shocking to most people when they really get close to the Warburg treatment is how well it alleviates pain, how quickly it can do that.
Let's wrap it up with if you had all the resources you wanted, anything you wanted, what would this look like?
Well, we need a TikTok video that go viral.
A different platform now. You can't do TikTok anymore.
Oh, okay. Sorry.
We need it taught in medical schools.
Yeah.
We need it to be sanctioned. That's sanctioning a concept that something can come out of us that could be taught in medical schools.
Well,
I'm sorry, my fault. Start over again. I need you guys to start with the statement. If we had all the resources we wanted, here's what we would do.
what I would like to do is to have.
You've built a lot of things. We've built an incredible groundwork, and now what's next to build on. If we had everything we needed, let's talk about what we could build.
Well,
If I were a rich
what
man.
I would like, what I would like is to have the government, the people, have more support for low-cost cancer treatments. We know the system that if you have a new drug, it costs so much to develop it and it takes so long that nothing is inexpensive by the time it's on the market. And yet we have something that doesn't need any new drug approval. We use 60-year-old drugs and very small doses. And how do you get that into the popular usage, right? And Mike talk about old drugs have a hard time getting redeveloped, and that path is not open. If this, again, people who are in charge of the system, in charge of making the system work for everybody, needs to help everybody, not just the few who can afford very expensive new drugs. And so maybe that's what I want is to have the Secretary of Health and Human Services put out a call to action for inexpensive treatment, and without having to go through 10 years of clinical trial.
But even in a simple non-inferiority trial showing that our particular protocol and regimen and compared to full dose chemotherapy, and is there a really huge difference? I actually don't think you'd see any superiority of the full dose chemotherapy side.
Well, I can tell you when people try full dose chemotherapy and did not work, and the cancer keep progressing and they're in pain, and then they switch to the Warburg treatment, more than 90% in my 16 years of observation will be much better. Will be, just pain goes away, can start eating again, get some appetite back, can rest, and all these quality of life things that palliative care, is supposed to deliver. And it's more than 90% is what I've seen.
But there is another major block, and that is the traditional attitude of oncologists. And if you think about how this all got started, this visionary went into hospitals and said, do you have anyone you can't help anymore? Do you have anyone you've given up on? Would you like to offer them this possible option? And that's one very important part, I think, about how this got started. By the legwork of going in, offering training for free in another country, because just try doing that here.
Yeah, I don't know how to do it here. Yeah,
That's why we went.
Yeah, So
Well, I think you're right. The only way is to just get the word out more.
yeah,
People would ask their doctors for this and know that it's available. If doctors knew a little bit more about possibilities,
Yeah.
you're not doing anything strange if you're using a 60-year-old drug.
yeah,
You know, I'm almost 60. I'm not the most interesting youngest thing in the room either, but I still have some value. So let's look at the drugs that have been around only because they're so well
Yeah.
established, so
Right.
well understood, so well characterized. It's frustrating to know that they're not being utilized to their potential.
right,
But the understanding that I have is that just the bias against non-traditional treatments is what would be relieved if there were, say, a course in medical school on nutrition, which usually there is not, or if there were a course on alternative methods that are scientifically sound. It would help keep oncologists from constantly saying, well, you didn't do clinical trials, so this idea is ridiculous. Or you haven't spent billions of dollars developing a drug, so how could you possibly get a silk purse out of a sow's ear? I think forcing or recommending courses that took this and or other optional treatments and made them be evaluated objectively would be useful in reforming the system.
Yeah. Well, I think, like you say, the system overall has some issues, and I think we lose sight as scientists and as doctors. We get focused on one thing and we expect that's the answer, when very often we can if we look at a system more holistically, we can see how every system affects everything else. And when we try to hit cancer with a sledgehammer, we don't have good results. But if we can tweak it in ways, like we said, that keep the immune system intact, nutritional elements that stay within the boundaries that we know are good for good health, there's a variety of things that we should all be doing to live and keep the body in a good state. 100 million years of evolution has built us in a way that is pretty good. And we don't need to make drastic changes. And the low doses, to me, just speaks to holistic elements where we want to make minor adjustments and let the body do some of the work instead of relying on the harshest and the most powerful chemistry.
Acute.
Yeah.
I don't mean to imply that there isn't a reason why the system has developed as it has. You have to keep people from making fraudulent offers. You have to have checks and balances. But I think we have checked and balanced ourselves out of the ability to recognize innovation when it occurs.
You want to wrap it up? Tom, you want to talk about if 5 years from now, 10 years from now, what do you imagine the United States treatment system is using in Warburg?
Well, I like to focus on the present because we're problem solvers in that when you ask a patient, what is your problem? You know, for late stage cancer patients, it's the pain. It's the pain. So we know we have the Right to Try Act. It's designed to help patients who have no other choice. And we, you know, as humans, we know that when someone is in pain, you got to help them. And the World Health Organization for palliative care, it says right there that use as much morphine as you need. In fact, you should probably be more aggressive in using morphine. And we know morphine doesn't kill cancer cells. And so I think if we focus on solving the problem of pain, cancer pain, we will get to a place where everybody can see the results. Number one, the safety, and number two, that it solves the problem of pain. And this is I think how this will become accepted by the mainstream oncologists, because every oncologist knows what happens when their patients are suffering from cancer pain. They don't have anything good that they can offer them. There's not that much they can do to help other than keep giving them doses of painkillers. And so I think that's how we start. And eventually we will get to a place where the National Cancer Institute, the American Cancer Society, somebody will say, well, let's see how well this works for earlier stages. And that would be a great place to be. And so if you ask me what I would like to see in 5 years is have a clinical trial comparing standard therapy and Warburg therapy in addition to whatever standard therapy, and look at the results. And that should be funded by, the government, I think, or somebody. And I think we can achieve that. But we have to start with solving the problem of cancer pain for people.